In an Augustinian abbey garden in the 1800s, a monk named Gregor Mendel observed the inheritance of specific traits in generations of pea plants. He used empirical analysis of quantitative data to reach conclusions that revolutionized the science of genetics. The basis of Mendel's discoveries arises from the fact that haploid sperm nuclei from pollen and haploid egg cells are formed by meiosis. When the sperm nucleus and the egg cell meet during fertilization, complementary alleles match up in new combinations. Mendel's model of inheritance rejected the homunculus and blending concepts postulated by other biologists in the 9th century. He showed that the breeding of tall pea plants with short or dwarf pea plants did not result in medium-tall plants. Instead, a fixed ratio of tall and dwarf plants resulted. He demonstrated that alternative forms of a gene combine and sort themselves out according to two simple fixed laws. We now understand that chromosomes consist of a sequence of genes. Alleles are two or more forms of a gene occupying a specific location on homologous chromosomes. Mendel's first law describes the segregation of alleles. When the chromosomes separate during meiosis I, the alleles are pulled to opposite poles. Later, Mendel found that the crossing of heterozygous F1 plants with more than one trait produces phenotypes in the F2 offspring in a ratio that is mathematically predictable only if traits are inherited independently. Mendel realized that the segregation of any one type of allele was not linked to any other. This segregation of alleles independent of one another was the only way that Mendel could explain the results of his F1 cross. It's now called the law of independent assortment.